The living world around us is based on innumerable chemical compounds.
Their characteristics and differences shape the interactions between living organisms and their environment.
These interactions are the main research focus of the Max Planck Institute for Chemical Ecology.
The institute was founded by the Max Planck Society in March 1996.
The town of Jena in Thuringia, home to the Friedrich Schiller University and other research centres, is an attractive scientific location.
The new building on the Beutenberg campus, situated close to other biological, physical and chemical institutes, offers excellent potential for scientific cooperation.
The institute attracts researchers from all over the world. Currently, it hosts scientists from 19 countries.
5 directors, more than 70 scientists and about 50 PhD and graduate students do their research here.
The research program of the institute is divided into 5 departments.
2 service groups support the main research directions.
Scientists in the Department of Molecular Ecology focus their research on the American plant species Nicotiana attenuata and the ubiquitous plant species Solanum nigrum.
In both of these plant systems, we have developed a series of tools that allow us to alter the expression of a gene that's important for an ecologic interaction.
The primary research objective in the department is to manipulate ecological interactions in nature in order to identify traits that are demonstrably important for the organism's Darwinian fitness.
One of the qualities of the tobacco model plant is that it produces nicotine, which is toxic to humans and insects.
Tobacco has the ability to produce nicotine on demand when attacked by herbivores.
How do the plants know when insects are eating them? What role does chemical signaling play?
These are questions the researchers are exploring, also by means of gene analysis and field trials.
The Department of Bioorganic Chemistry has 2 main research areas.
The first one is our interest in plant-insect interactions, and there we want to understand how insects trigger the defense reactions of plants.
When attacked, plants emit volatile substances. These compounds not only lure predatory insects that feed on the pest species,
they also induce defense reactions in neighboring unattacked conspecifics.
The second part of the research interest explores the defenses of the insects.
How do insects use the substances in the plant tissue?
How are these substances transported?
How are they chemically transformed into defense compounds by the insects?
Why is there such a vast variety of defense molecules?
What are the functions of individual compounds in plant defense against herbivores?
We care about genes, we care about proteins, we care about enzymes and metabolites and how they interact in making defensive compounds in plants and in, on the herbivore side, how they are involved in detoxification or processing defenses.
Different components have different roles in the mechanisms of how plants defend themselves and how herbivores respond to these defenses.
For example, scientists in the department are searching for answers to these questions by studying the formation of glucosinolates, or volatile terpenes.
The biochemists are interested in how these plant substances are produced and how they accumulate in the plant tissues after herbivore attack.
They are also investigating genetic aspects of the secondary metabolism in Arabidopsis thaliana and related species, as well as in spruce, corn and cotton.
Another two departments focus their research on the role of insects in chemical ecology. Insects are masters in detecting volatile chemical signals, even at extremely low concentrations. The insect's nose is its antenna. Different odors can act as attractants or repellents. How does the insect antenna detect such compounds?
We're investigating odor perception in insects, how molecules move from antennal receptor neurons to the brain, from molecule to mind. But we're also looking at the behavior of these insects and how they will move to an odor or how they will go away from an odor.
Using modern two-photon laser scanning microscopes and ion-specific tracer molecules, the scientists are able to observe how olfactory information is processed in insect brains. Molecular cloning of genes encoding antennal receptor proteins is the basis for understanding their structure and function. Receptor genes are heterologously expressed, for example, in human embryonic kidney cells. Patch clamp recording and optical imaging allow measurements of electrical and chemical changes after stimulation with selected odor compounds. Several experiments are performed to study the behavior of insects under different environmental conditions. By combining analyses of receptors, nerves and brains with behavioral studies, we can gain new detailed insights into the interactions between insects and their environment.
The chemical aspects of plant-insect interactions, studied from the insect's perspective, comprise the central research topic of the newly established Department of Entomology.
In the Department of Entomology, we are interested in how insects can overcome toxic compounds that plants make to deter their herbivory.
The different questions posed by the entomologists require broad analytical know-how.
The primary focus is on herbivorous Lepidoptera, although other systems are also being explored.
NMR Working Group is one of two groups working on analytical tasks.
The purpose of our research group is to do service for the other departments in the Institute, for running NMR spectra, for identification of natural products and for synthetic compounds.
The NMR group investigates the metabolic processes of plants by means of C13 and N15 NMR spectroscopy.
Research focuses on studies of the metabolism of brasinosteroids, a new class of plant hormones.
And of phenylphenolanons, unusual plant pigments.
The second group in the Institute is working on decoding the structure of chemical substances.
We identify organic compounds by means of mass spectrometry. We are mostly interested in compounds which mediate communication in insect and plant world.
Experts from the mass spectrometry group use innovative analytical techniques.
They conduct experiments to investigate plant and insect signaling substances and how these substances bind transport proteins such as lipocalins and PBPs.
The Institute has established a joint program with the university and other research centres, offering excellent research opportunities and education to junior scientists.
In addition to research work, the students get good supervision by 2 supervisors. They have additional courses and seminars, and they get to know each other very well by joint retreats.
The most important goal of this interaction with the Max Planck Institute is to have access to different equipment and also the different methods which are established in different laboratories.
In 2001, the Max Planck Institute for Chemical Ecology moved into a large new building, in itself a fine piece of modern architecture.
Hello.
The Max Planck Institute for Chemical Ecology regularly opens its doors to the public.
The Max Planck Institute for Chemical Ecology expects that the results of its basic research will also attract farmers and agricultural scientists.
Growers and plant breeders who know which mechanisms are involved in certain plant-insect interactions can use these insights to produce new cultivars.
Jena, a town with a great educational and industrial tradition, today offers excellent conditions for groundbreaking research.
First-class educational opportunities,
plenty of services to meet a wide variety of professional
and personal needs and interests,
and a pleasant atmosphere.
The Max Planck Institute for Chemical Ecology is convinced that work done here will enormously influence modern agriculture throughout Europe
and all over the world.